Welcome everyone to the Bentley Lecture. We'll be getting started right away. We'd like to remind you that this session is being recorded. We will have the Q&A function available to you throughout the presentation. Welcome.
Thanks, Christine. This is my chance to welcome everyone to the 2022 Bentley Lecture on Sustainable Agriculture. My name is Brent Swallow, and I'm a professor in Resource Economics and Environmental Sociology in the Faculty of Agricultural Life and Environmental Sciences here at the University of Alberta. I'm pleased to now welcome the Dean of our Faculty of Agricultural Life and Environmental Sciences, Dr. Stan Blade.
Well, good afternoon, everyone. Good evening, good morning. I know that we have people joining us from across the globe. It's great to have you all with us for the Bentley Lecture. Before we begin, I would just like to acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. I would also like to acknowledge that I know that we have a couple of Dr. Bentley's children registered, both Ann Mason and Ted Bentley had registered to join us, and certainly welcome to you and perhaps even to a grandchild or two that are joining us as well. I'd like to tell you a little bit about the Bentley Lecture. Professor Fred Bentley, his academic career was mainly at the University of Alberta, spanning 36 years, serving as the dean of our faculty for 10 years from 1958 to 1968. Dr. Bentley just had an extraordinary influence, not only here in Alberta, but across Canada and worldwide. He was the recipient of the Alberta Order of Excellence, the Order of Canada, served as the chair and board member of ICRISAT for many years. So just this remarkable influence in the area, not only of soil science, but of agriculture and food. We're so pleased that we are able to host the Bentley Lecture in Sustainable Agriculture. There was an endowment fund that was created in Professor Bentley's name so that each year we can invite a speaker of international renown to present current research and updates in areas related to soil science, agricultural production methods, and sustainable agriculture. And as you have already seen from Dr. Winowiecki's bio, our presenter today certainly captures that thought, and we're very pleased to welcome all of you today to participate in this Bentley Lecture. So welcome once again. Thank you to our sponsors that have been so supportive over the years, and for all of you that are joining us and taking time out of your schedules. And with that, I'll hand things back to Brent.
Thank you very much, Stan. Just on your very last point there, so I'm going to talk about some housekeeping things. The first one relates to your last point about the endowment fund and how much we value the contributions that have been made over the years. So thank you to all of your organizations, and we'll be back to you again when we get back into live sessions, which we hope will be next year. Just to let you all know that we now have 152 participants, so we've got a nice audience, so welcome everyone. Our guest speaker's presentation today will last about 40 minutes, and we invite you to ask your questions during the presentation, please using the Q&A function rather than the chat. Please don't use chat, let's just use Q&A so we can keep track of the questions. So as Dr. Winowinecki is going through her presentation, please just note your questions. You can type them there. We expect about 20 minutes after the 40-minute presentation, about 20 minutes for a question and answer period. And then we will break and thank Dr. Winowinecki for her presentation and move into another session we have, which is going to be presentations by students. So they will be presenting research to you in the style of the 3-minute thesis. So quick kind of lightning presentations. We really hope that you'll support our students by staying a bit longer to hear about the interesting research that they're all doing. That will be about 35 minutes or so. Also joining me as hosts today are my colleagues Nadir Erbeglin chair of the Department of Renewable Resources and professor in forest entomology and chemical ecology, and Guillermo Hernandez Ramirez, associate professor and lead of the Sustainable Land Ecosystems Research Group. You will get to meet both of them a little later in the presentation. So now I have the great honor to introduce Leanne to you. And to set that up, I just want to give you a little, just remind you of some facts. The Food and Agriculture Organization now estimates that land degradation affects about 2 billion hectares of land that is home to 1.5 billion people. So that's more than 1 out of 10 of us, 1 out of 10 acres of arable land on the planet, and something like 20% of the world's population. Despite strong consensus on its urgency and importance, progress in countering land degradation processes remains slow. Fortunately, however, there are real opportunities to slow and reverse land degradation processes, and there are determined scientists. Dr. Leigh Ann Winowiecki is one of the most determined, prolific, and optimistic of these scientists. She is a most fitting scholar to deliver this year's Bentley Lecture in Sustainable Agriculture. Dr. Leigh Ann Winowiecki is a soil systems scientist and research lead at the World Agroforestry Center. She's based in Nairobi, Kenya, where she's coming to us from tonight for her, but works across the tropical regions of the world. Leigh Ann earned her PhD in soil science and tropical agroforestry through a joint doctoral program at the University of Idaho and Cartier in Costa Rica, and was a postdoc at Columbia University in New York. She joined the World Agroforestry Center, ICRAF, in 2009, which happened to be exactly the same time that I left that same organization. So I guess ICRAF wasn't big enough for both of us. Leigh has played a critical role in developing and implementing ICRAF's land degradation surveillance framework, a systematic monitoring framework for assessing land and soil health that has been implemented in over 40 countries with many, many partners. She has published widely on soil organic carbon, ecosystem services, and land degradation across Africa and many other countries. She currently leads a major international initiative on restoration of degraded lands in East Africa and the Sahel regions of Africa, as well as the Coalition of Action for Soil Health that was inspired by the 2021 UN Food Systems Summit. So with that, I'll hand you over to Leanne.
Wow, thank you so much for that introduction, Brent, and so nice to be here and to meet everyone. Yes, I'm Leigh Winowiecki, and I just need you to stop sharing screen so that I can share my screen. Great, so I hope everyone can see me, and I will get started right away. Today, I'm going to talk about advancements in landscape scale soil monitoring, for carbon accounting, and for tracking restoration. And I was getting into my presentation and I thought, oh, okay, what would I want the audience to take away? So I just want to offer my key messages right at the beginning, especially if people have to leave early. So one is that there's this real need to connect science and farming. And that as scientists, we have a very important role to use and translate science into action to inform decisions. These can be decisions at the farm scale, national level, intervention level. And today I'm really going to talk about the tools and methods that we have to track and measure soil and land health, as well as underlying processes of degradation. And it's not going to be all doom and gloom, because I want the key message that through stewardship, we can improve soil health. And that one of the ways to do this is through encouraging farmer innovation. So making sure we tailor interventions to meet farmers' needs, his or her needs, and that a real call for public-private sector financing and engagement to help us overcome the barriers currently. And as we dive into the presentation, I thought nobody knows who I am. And Brent has done such a great job giving some background, but I just thought I'm new to this community. Let me just share quickly some of my background. So I am your neighbor. I'm from the US and I'm actually from Detroit, Michigan. So always close to the Canadian border. And I did my bachelor's in chemistry, my master's in soil chemistry, and my PhD in Costa Rica in soil pedology. And it was a dual PhD in agroforestry. And as Brent said, I started my postdoc at Columbia University, but that really brought me to Africa. So I was based in Tanzania and basically spent a lot of time in the field in 5 countries in East Africa. Then I joined the CGIAR in 2011. And now I am a global theme leader of soil and land health for C4E Craft. So I thought it was important, since we have so many students, to share our experiences. Of course, they're all different. And a little bit more about me. I took these photos, except for the one on the bottom left, of my garden here. So I have this saying that you should never trust a soil scientist unless they compost or try to grow things. So I'm demonstrating my organic garden in the top left. These are my chickens. This is my compost from the 24 cows. And this is me in the fields with my students earlier this year in a rangeland. So now you know so much about me. I hope to learn so much about you. So why are we here? So there's a global relevance of soil. Over 95% of our food is produced in soil. And through improved management, we can actually produce more nutritious food. And soil has many functions. And this is why there's so much to study around soil science. Soil delivers multiple ecosystem services. I'm sure we know that 80% of the carbon stored in terrestrial ecosystems is actually in the soil. So we have a huge potential to store soil carbon in the soil, contributing to climate change mitigation. Soil is also important for nutrient cycling. It's important for flood regulation, water cycling. So I really like this graphic because actually there's many PhDs just in one of these pieces of the pie. And of course, having a soil-centric focus, I really think that healthy soil is central to achieving so many of the Sustainable Development Goals. So you can see here everything from gender to life on land to climate action and zero hunger. And as Brent opened up, more than 25%, and actually estimates are up to 40% of the Earth's surface is degraded. It's impacting 3.2 billion people. This is a landscape that we took a photo of using a drone in eastern Kenya. You can really see the lack of soil cover. But I thought I would just at least give one slide of definitions. So when we say degradation, what are we talking about? And I like this definition because it talks about land degradation is when the ecosystem, the land, is no longer to provide the services or the functions. So remember the slide of the soil ecosystem services provided by soil. Degraded soil would no longer be able to perform all of those services. And in terms of soil, we have many different types of degradation. We have chemical degradation, where I'm showing here a photo I took of a highly salinized soil. So you can see the salt crusting on the surface. We have physical degradation, so that's compaction when water cannot enter, or erosion, and then biological. We are really working hard this year with the Convention on Biological Diversity to finally start including soil biology in their declaration, because soil hosts so much diversity underground. So this is a photo taken by our son in Zambia. So yes, even on our vacation, we are taking degradation photos, but to demonstrate that soil erosion is one of the most widespread forms of degradation. And I like to remind us that degradation is usually not just the consequence of one action, one point in time, but instead it's a result of diverse and mutually reinforcing actions, including anthropogenic and natural drivers. So I know that it will be impossible for you to believe, but this is actually was actually an agricultural landscape. The up above that tree, farmers were actually trying to grow crops there underneath some of the native vegetation. And when we look here, I'm just asking, what are the forms of degradation you see? So we see some erosion. Not sure if you can see some of the calcium carbonate deposits on top of the soil, but also we see some vegetation here, the Sansevieria. It is indigenous, but it's aggressive and it's starting to take over this area. So there's many forms of degradation. And unfortunately, I took this photo only 2 weekends ago in one of our field sites. And this is because in Africa, the estimates are that over 65% of the land is degraded. So now let's talk about the positive. So I think I've made the case that investments in soil health practices are urgently needed. They're needed to reverse and prevent land and soil degradation. They're needed because soil needs to provide nutritious food. No longer can we just say food, it needs to be nutritious, contribute to climate change, realize the Sustainable Development Goals. And I hope everyone here is aware that the UN Decade on Ecosystem Restoration was launched on June 5th last year. So we can, by restoring soil health, we can also reach our ecosystem restoration targets. But looking at this landscape, which is actually in Machakos County in eastern Kenya, you see that landscapes are diverse. This is another landscape in southern Tanzania. Landscapes are diverse. The scale of degradation is massive. So how can we start tackling degradation through restoration, through on-farm restoration, when there's so much variability and so much complexity? And I think it's important that when we design studies and when we're implementing on the ground, that we really need methodologies that allow us to understand the context and this variability. And if we're developing and implementing a sampling design, that sampling design must capture this variability in order to prioritize the right interventions. But then if we're sampling landscapes, that means that we will need to use soil analysis and technologies that are actually low cost and robust. And these frameworks then need to track changes over time across this variability. So at eCraft, and now we're merged with C4, so we say C4 eCraft, we've really been doing this for the last 15, 20 years. So we're combining multiple innovative monitoring techniques to better target and understand processes, but also track restoration in real time. That's the other key. We can no longer take 5 years to do a baseline study. Well, we never should have in the first place. So we're combining remote sensing, and I'm not sure why my remote sensing diagram is focusing on the people, but I think you can imagine that it's shining on the land behind them. We also have a systematic land degradation surveillance framework. And then we've also been embracing assisted citizen science, where farmers, lead farmers, NGOs are also engaging in this monitoring practice. So that's what I'm going to focus on today. And to do that, we have to understand what indicators we're going to use. Now, I'm sure everyone in this room can debate for weeks on end of which indicators, but please allow me to share these 5 principles that we use when we're deciding on our indicators. They're science-based. They need to be readily measurable, so that's quantifiable. Remember, we cannot take 5 years. It needs to be rapid. It needs to be based on multiple scales and represent complex processes across the landscapes. So just think in your mind, what are some of the key indicators that you think are important to assess degradation, track restoration? And remember, we want them to be consistent, so across various ecosystems. So I hope everyone has one in their mind, and then let's see if they match up with what we measure in the LDSF. Again, I'm showing you a photo of my garden because soil organic carbon is one of those really nice indicators for soil health. Why is it nice? Why do I think it's nice? Because it influences many other processes. So with higher carbon, you should have higher water holding capacity of the soil. You have better cation exchange capacity, and we know it can positively influence land and agricultural productivity. But Also, it responds to management. So if you do not take care of your soil, then you will be decreasing the organic carbon. While if you're replenishing your soil, it will increase carbon. Also, it's quantifiable, rapid to measure, and we can map it. But just a reminder, it's not the only indicator, but it is a key indicator. So the frameworks that we use should have multiple indicators simultaneously. So in the LDSF, these are the variables that we measure. So I hope that this is an overlap with what some of the participants were thinking. Multiple soil health variables, including biological indicators. We also look at degradation around erosion, root depth restrictions, soil hydrology, looking at saturated hydraulic conductivity, of course, vegetation assessments, trees, shrubs, rangeland health, grasses, but also looking at land management. So what are the agricultural practices? And I work at World Agroforestry. We love trees. I think you can see from the photo in the background from northern Kenya that it's not just about one thing. It's not just about trees. We still have erosion in this landscape because there's a lack of vegetation covering the soil. So that's why we need multiple indicators. So the LDSF was developed. It's a systematic assessment. It assesses variability across the landscape. This huge data set that it collects allows us to conduct statistical analysis. So I know now everyone's asleep because I said the word statistics. And in general, soil scientists do not love statistics, but we're a new breed, right? And we have new training in big data. So now we have to be good at statistics. And then this also allows us to produce high quality maps and assess changes over time. But the power is first, of course, the indicators, but that this network has been implemented in over 43 countries. Now, obviously, there's a huge bias here in Africa, and we are slowly removing that bias and extending more sites outside of Africa, but our headquarters are there, and we really started implementing this there. And I show these photos because remember, these are all field-based sampling places. So here is Providence in Rwanda on the left. We have Senegal, Lesotho, India, and Vietnam. So all of these dots represent a 100 square kilometer sampling site. When we have this network of systematic sampling sites, the world is endless of possibilities for the analysis that we can do, the questions we can answer, and anyone who's inspired by anything I show today, please reach out so we can work together. So here's the sampling scheme. Each one of those dots is a 10 kilometer by 10 kilometer by 10 kilometer landscape. And I hope you can see the white dots here. So this sampling design is completely, well, it's stratified random. So it's unbiased. So this is the other unique aspect. Unbiased sampling encourages more robust statistical analysis. So we have 16 clusters. You can see how the white dots are clustered, and each cluster is 1 square kilometer. I like showing these because just in this landscape, you can see how when you randomize 10 plots, you're capturing a lot of the variability that exists in the landscape. Then all the action happens at the plot level. That's all the observations. The soil sampling is right here at the plot and subplot level on the bottom right. So that's the nitty gritty. Anyone who's really good at math here sees that this is an exponential sampling design from 100 meters square subplots, 1,000 meter square plots to 1 kilometer square clusters, and so on. So you can imagine the type of mixed effects modeling that we can do. We collect a lot of soil samples. So here's some photos from the field, a lot, 160 topsoil, 160 subsoil, and then instead of bulk density, we do cumulative mass. Now, all the soil scientists in the room are cringing because they're thinking, oh my, we can't afford to analyze 320 top and sub samples per site using traditional wet chemistry. And you're absolutely right. This methodology is very much based on a new technology, well, new 20 years, called soil spectroscopy. It uses light to analyze soil. And in fact, we use mid-infrared light. We analyze all the samples on spectroscopy at $4 a sample. And then we only analyze 10% of the samples on wet chemistry. And this allows us to build spectral libraries to predict soil properties with high accuracy. Okay, and we call it, it's very simple, shining a light on soil. So this is Elvis in our lab. This is one of the mobile spectrometers, the Alpha 2 MIR. And like I said, now that we're changing how we analyze soils, this is also transforming research, and it's requiring new skills for soil scientists. So now we're getting spectra every time we analyze the soil, which requires expertise in database management, data management, statistics, and really shaping and reshaping soil scientists of the future. And eCraft has invested the last 20 years to build this consistent library, and we support over 43 labs globally. And we just submitted a proposal this morning to bring Europe up to where Africa is in terms of scaling spectroscopy. It's very accurate. So on the right are the measured versus predicted SOC values for a paper that we did in South Africa. And you can see we're getting R squares of over 0.94 for pH, carbon, sand, clay, also for nitrogen exchangeable bases. And again, this is Dickens on the top left on our brand new mothership, the Invenio MIR, which analyzes 600 samples a day, 600 soil samples a day. So enough about that. Let's inspire with some applications of the LDSF, some outputs. I hope everyone is still with me. So one of the things that we really are interested in, as I mentioned, is how does soil organic carbon vary across the landscape? So here are 4 LDSF sites, 4, all in East Africa. And even if you don't know what the colors mean or where they're from, I think we can all agree that the spatial patterns are completely different in all landscapes. I will tell you that yellow is lower carbon. So in the bottom left, that's Kipsing, just going into the Laikipia drylands in central Kenya. And the top is southwestern Tanzania and Mbinga, where there is actually higher carbon. So we can map carbon, and we can also calculate carbon stock. So we can look at carbon climate change mitigation potential in these landscapes. And here's the violin plots. We also did further analysis and found that in eroded plots, carbon stocks were lower in all sites. So whenever someone asks me, probably someone will ask me in the chat, what can we do to increase carbon? I say the first thing we have to do is stop erosion. We need to curb, stop, combat soil erosion. Then we start building the soil with our interventions. And that's what our research has been showing over and over again. The erosion is prevalent, and it's really prohibiting us from storing more carbon in the soil. So this is one paper from 2013. But I wanted to take a pause. So I'm not sure if you can see the pause button here, because wait, soil mapping has been going on forever. What is the difference between the map I just showed, which is actually at 30-meter resolution, and the traditional polygon maps, which I think people may be more comfortable with here? Well, one of the things is that these types of maps are mapping functional soil properties, carbon, pH, for example, and they can be used to track changes over time. The soil polygon maps are very different. They don't so much capture the variation of the soil functional properties, but they provide other meaningful information. The other thing is that with the LDSF data, we combine the field data with the remote sensing to produce these maps, and they can be produced at whatever scale the landowner or land user wants. So depending on the satellite or the sensor, 5 meters, 10 meters, 30 meters, 500 meters. So that was our pause. There will be a quiz at the end for the students on the difference of these maps. So more maps. So Tanzania is basically where I did my postdoc. And so we produced these soil carbon stock maps for all of Tanzania. Yellow again is low, brown is high. And we didn't just stop there. We also looked at across these 7 LDSF sites, how do the soil carbon stocks vary by site, by topsoil, by subsoil, and in cultivated land versus non-cultivated land. And when you have these big data sets, you can see sites that really have a stronger effective cultivation, like in Mbinga and Bukwaya and Chinyanguku. And we can start piecing together the stories about what's happening at the landscape and how cultivation and management practices are influencing carbon and erosion, for example. We can also look at relationships between soil variables. So I know I'm talking a lot about carbon, but we have to acknowledge that there are inherent soil properties that will ultimately provide constraint envelopes of how much carbon we can actually store. So sand, for example, which is an inherent soil property dependent on the parent material and the weathering, a sandier soil will just inherently store less carbon. And that's actually what our data is showing here. So on the y-axis is soil organic carbon, on the x-axis is sand content. And you can see Mbinga is this purple site where I showed you before, has high variability in carbon, higher carbon, but very little variability in sand. While some of the other sites are really in the sandy side and have low carbon. Also, we looked at pH and exchangeable bases, SOC, and TN. And this is some of the real paradigm shifts in soil science because our loved textbooks are now starting to include some of these big data sets and talking about how data is shaping our theories around soil science. And I'll point out, this is over 1,000 soil samples in these graphs. More maps of pH. I show this one because we use the soil data from the field, from the georeference locations of the sampling to produce these maps with the remote sensing. And you can see here in a 5-meter resolution, the variation in pH with higher pH on the right and lower pH on the left. And this is actually due to geology, but we did not include a geologic layer in the map, but we're still able to pick this up because of the amount of data in the reflectance data from the satellite imagery. And then big scale again. So this is the root depth restrictions across the African continent at 500-meter resolution. And we can also look at vegetation changes. So I really hope that all of the students here, and actually all of us in the next few years, we can actually start seeing reversed trends. We can start saying degradation is actually lower and vegetation cover if indigenous vegetation is higher. But this is an example of how 1990, and this is made by Tor Vaughan, in 2000, 2017, the areas of green, so the higher fractional vegetation cover is decreasing, and the areas of brown are actually increasing. And I show this because these are project areas where we're actively implementing restoration with farmer managed natural regeneration. And we're tracking now from 2017 to now, what effect our interventions are having on vegetation cover. And this area in Ghana is a nice example where the trend has been decreasing over time. And now with the UN Decade of Ecosystem Restoration, let's hope our data will start to see an increase. Also, I show this not to make you dizzy, but in each LDSF site, we also look at land use category. I think sometimes we forget how dynamic landscapes are. Each color represents whether it's an annual crop with agroforestry or an annual crop with no trees, annual crop or perennial crop like tea with trees or coffee with trees, pasture rangeland. And you can see each one of these 100 square kilometer sites has so much variability. And it's just a reminder that when we're working with smallholder farmers, that these landscapes can change. It can go from being an annual agroforestry to annual crop once they cut down the trees or go into fallow or even go into rangeland. So when we're conducting studies and surveys, it's really important to acknowledge this variability. I have to show some data on trees because they're talking so much about soil. So we look at all the trees on the landscape. And this is an example. We actually published this in earlier this year. And this is in Rwanda. So we looked at plots, the 160 plots that were not cultivated and cultivated. And then on the x-axis is the species. And I hope you can see them. If not, I've pointed it out in the boxes. So the most common species were non-indigenous. So this is Rwanda. Anyone who knows Rwanda, this is a real issue and something that our projects are looking at. So eucalyptus and grevillea were there. But then you see in the cultivated plots that there were, so the higher the bar, the more frequent these trees were in the landscape, that there are some nice citrus and mango trees planted on the farm. So farmers are planting trees. And then over here in the very low numbers are some indigenous species that were only found in non-cultivated areas. And we're just gathering so much information. So how can we make sure that that seed stock is not lost, that we can start nudging farmers to incorporate some of these nitrogen fixing trees, in fact, into the farmland to improve soil fertility? So now back to some maps. So we started off talking about erosion, and I just want to end on a few things and bring it to the farmer. So this is the global assessment we published in 2019 using LDSF data on erosion. Hot, red, and yellow are the hot spots of erosion. And we really think that now that we've made these data and maps for multiple years available, and I can share that link on the landscape portal, these are put together by Torvagen, that now we can start including them in climate assessments, crop modeling, and acknowledge the important role that erosion and degradation has for climate change vulnerability, for crop productivity. We've also done this at national level. And as you may or may not know, Kenya has committed to restore 5.1 million hectares. So let's use some of these erosion maps and target where to do the interventions. But remember, where are these interventions going to take place? They're actually going to take place on farmers' fields by farmers. So we call this farmer-centered land restoration. This is one of our farmers, not ours, one of the wonderful farmers we work with, where he's demonstrating the productivity of his maize in these soil water conservation basins with manure and with trees in agroforestry versus traditional farmer practice. Each one of these red dots is actually georeferenced farmer locations where we've been actually working with them for the past 7 years on landscape restoration on farm. And we're tracking how their interventions are influencing, hopefully stopping soil erosion, as well as soil carbon. And Brent, we are so pleased that we analyzed 3,920 soil samples funded by WLE and found that across this 3 counties, and after 4 years of intervention, we're actually increasing soil carbon. So really fantastic news here in Eastern Kenya. So we're also using this in rangelands to prioritize rangelands restoration. So we have soil carbon plus erosion, and then we do restoration priority mapping. And we have a bunch of papers and blogs on this, but most importantly is that this is a conservation. I know it looks like a weird Lego guy, but it's a conservation area where they're now implementing restoration and prioritizing their interventions based on some of these maps. And in Regreening Africa, we are trying to restore 1 million hectares. So we're using the land degradation surveillance framework to set the baseline, but now we're doing assisted citizen science. This can be used in Canada. It's called the Regreening Africa app. It's free on Google Play Store. And this app is now putting the monitoring in the hands of the implementers. And currently has 4 modules. Version 2 is coming out any day now. So we're looking at tree planting, FMNR, nurseries, and training. You can guess the next new module will be something around soil water conservation. And the idea is that farmers walk around their plot, and here's a polygon you can see on the screen. they walk around there and then they register which trees are there. Are they farmer managed, natural regeneration, tree planting, survival rate? What's the use of the tree? And what we've done is we've put this all online on a dashboard so you can look at species and how the different interventions are really complementing species composition across the landscape. We have over 2 million trees that have been tagged in 8 countries using this app. It's absolutely amazing. So if you're interested in tree planting or FMNR restoration projects, you can use this for monitoring. And as I said, also looking at where farmers are planting these trees and how they're using it: firewood, construction, medicinal. So I think I have about 3 minutes left, and I just want to talk about one of my key messages I mentioned, which is about translating science into action. So these maps and data are so great, but it's really about working with farmers because they're the land stewards, and that's how we're going to reach these restoration goals. So these tools that we're using are to engage farmers in the monitoring, have co-learning with indigenous knowledge and scientific knowledge. And what we found is that this co-monitoring together is actually leading to further scaling and further implementation on the ground. So this is a really cool Mongo-based story we had on our sites in Eastern Kenya, linking agroforestry and the Regreening Africa app. And here are some of the interventions. So you'll see covering the soil, planting fast-growing papaya trees first to get some nice benefits, and then some slower growing trees in between, so that the farmers are receiving benefits from the papayas while they're waiting for the benefits from the other trees. And also talking about women's empowerment. So we do these agroforestry basins where we're putting compost and manure and having these basins where they're planting their crops. And we found that women are really kind of organizing, as women do, to work together and scale this. And so we have quite a big program on impact of land restoration on the agency of women, acknowledging intra-household dynamics, male migration, and aspirations. And the final point is that I think we've talked a lot about variability. And so I want to leave you with this message that there is no one silver bullet. Unfortunately, there's no one silver bullet that works for every context. And it's really about empowering and encouraging farmers, all of us, to innovate for what works for each farmer. So that's experimentation to answer questions that farmers may have. And this requires a shift in behavior from researchers. I've said this many times already. We cannot take 5 years to deliver the data. We cannot just analyze soil samples and do the monitoring without including the implementers and the farmers. So there's also a shift in behavior of donors, development actors, and farmers to really have this co-learning across all these communities of practice. And this also requires that we put the data out, open access. This is an example of one of the Makoweni County Resource Hub that was co-designed with the county. All those maps, all these data are available online for free. And then really bringing the global community together for the scaling of soil health. So out of the UN Food System Summit, we've developed a coalition of action for soil health. We'd love to have University of Alberta there. We have 7 member states, 50 organizations, international research organizations, NGOs, 10 private sector companies signing on to really work together to scale soil health globally. We need to overcome the barriers of monitoring, implementation, and policy. And that's what we'll focus on. So back to our key messages: we can improve soil health with stewardship. We need to scale these investments urgently. We need these coalitions, we need farmer innovation, and that we do have the tools and methods to translate science into action. I hope I've not gone over time, and I hope everyone is still awake. Thank you.
Thank you very much, Dr. Leigh. I really appreciate it. That say your enlightening lecture on I tried to keep up some of the keywords that you use: the soil and land degradation, soil health, landscape restoration, and the land degradation surveillance framework. These are really, really impressive keywords in a presentation spread out and very meaningful. You emphasized several times the take-home messages. As I have my own take-home messages. Some are resemble to yours. One of them is, as a scientist, I think we have obligation to translate science into action to help communities as well as to solve some emerging problems. I think that's really, really important message from your presentation lecture, as well as your last point about the stewardship, right? It's a must, to improve soil health as well as to stop land degradation. That is sort of my take-home messages. Now I'm looking at the questions. We have about 15 minutes. I really appreciate it. So questions are already there. One of the questions is about the use of biochar, right, at the small scales, whether is a way of mitigating climate change, whether that can be integrated in your framework. That's the question from Rob.
Great. Thank you so much, Nadir. And thank you, Rob. Yes, so there are a lot of really interesting studies going on with biochar and even how to use waste. So in Western Kenya, there's a lot of sugarcane production, and there's some local studies on converting the sugarcane waste into biochar. And I think that there's been some really important studies on the benefits or, the use of biochar for agricultural productivity, and absolutely can be included within an LDSF site. We have a lot of intervention sites that are included. And so I think including biochar in there would be very interesting. Absolutely. I also saw a question about how we measure soil carbon. And yes, I agree. So we really we calibrate all of our spectra with dry combustion. And I think right now, especially with the carbon markets and the need to track changes over time, we must make sure we are accurate and robust. And so we analyze all of our samples using spectroscopy, and then 10% using dry combustion, so that we get the most accurate gold standard method for carbon. And then we found that we're actually able to track changes over time, because the accuracy of this method is so good that we can even map very low carbon environments. That's what we were doing this evening, waiting for this lecture, looking at Eastern India and Andhra Pradesh, where you can't imagine is 5 grams per kilogram carbon, very, very low.
Thank you. And another question from Dale is, what motivates your cooperators to take a part in this rather very interesting study?
Actually, it's so amazing. So when we were developing the land degradation surveillance framework, like, oh yeah, if we get 5 sites, 10 sites, 20, 100 sites, that would be great. But now we have over 200 sites, and NGOs, universities, students, development projects, research projects, donors, many donors are using it. And I think it's because using the LDSF is really robust, but we can basically do 100 square kilometer site in 16 days. So my team is just coming back from Uganda. They just finished 2 sites there. So already this year we've done 5 sites. So it's really, we're able to get in the ground. They put everything in electronic data entry. They bring the samples here, 600 samples a day we can analyze on MIR spectroscopy. So it's really, that turnaround time is low, much faster. And the other thing that's motivating, I think now we're in the big data age. We really are in the big data age. And that's why I was stressing these new tools using R or Python, these data analytics. And we want data. Knowledge is power, whether you're a scientist or a farmer or a government. So we really talk about evidence-based policy. So now how can the Kenyan government use these maps in their nationally determined contribution?
Thank you. So there is a comment by the anonymous attendees suggesting basically saying that the listener basically found it very interesting that farmers implementing solutions on their lands. So there is a similar project done in Canada about the farmers that they're leading a project that basically protecting the agricultural land. Questions by Luis suggesting that could you go back over the technology you're using to measure soil organic carbon?
Hopefully, I think I covered it, but yes, so we're basically analyzing all the soil samples using spectroscopy. So that's just shining light, and then we get a spectra, which we call the fingerprint of the soil, and we can actually measure multiple indicators simultaneously. We can look at soil condition, but also soil carbon. And then 10% of the samples go to get analyzed by dry combustion. We also do a lot on isotopes. And I saw there was a question there on vegetation from John. Can I take that one? Because I thought that was interesting.
Go for it, please.
So talking about soil carbon and vegetation, and what sorts of vegetation would we want to encourage in the sites and the different management regimes. I love that question because we don't even, on our form, we call it semi-natural vegetation because I'm not convinced how much undisturbed vegetation we have left here. So we know that so much of the landscape has been impacted through management or use. So one of the principles that we have is, vegetation at all levels. So covering the soil, shrub layers, and then, multi-strata agroforestry systems to the trees. And really encouraging this multi-strata is so important because each one of these layers has such an important role for healthy ecosystems. And we also want to make sure we're never spreading invasive species, especially ones that are on the IUCN Red List. I know it seems obvious, but it's not always obvious. And there's many examples. Brent can tell you decades of examples of wrong plantings in Baringo. And then, we work with farmers to understand what they need. Are they looking for timber? Are they looking for fruit trees? And so making sure that we have this mix of indigenous species plus high value tree species. So that's what eCraft is all about and what we've been doing for many, many years. Thanks.
Thank you very much. It's another comment from Julie suggesting that, enjoy the presentations, and she's asking whether can or how we can utilize your data about integrating and putting the farmers on charge and using this one to show the world that, this is a successful example.
Yes, and I think that's a fabulous idea. And actually, that's one of the main goals of the Coalition of Action for Soil Health. And anyone who's interested should reach out, and I can put you can share my email. And it's really about having this co-learning across countries, across ecosystems. And as I mentioned, we're really strong on how we can use the data. Use these data sets to inform the decisions, inform the policy. And so this can be from anywhere. And we have to recognize the important role of farmers, pastoralists, forest managers, who are really the stewards of the land and will be the ones restoring the earth. And so I love working with, I think we have to all come together, scientists, farmers, policymakers, research, everyone needs to be in the room talking.
Thank you very much. There's another question from Ross is asking whether there is going to be a role of sociologists or sociology for facilitating the transition from science to farmer applications in soil research. Whether they have a role.
Yes, and anyone who wants to work with us, we're obviously very biophysically based. We have a few papers on socio-ecological systems and really co-locating. Now we almost co-locate all of our sites with household surveys, socioeconomists, gender experts, because we can't do it alone. We have to work together in interdisciplinary teams. In fact, my PhD was in an interdisciplinary team of 7 PhD students. Is a wonderful, I presented my research, which is very biophysically based. So please reach out. There's a huge, important role.
Thank you very much. It's another question from Lisa, whether you mentioned about the soil microbes in your presentations, and she's wondering whether how do you measure these soil microbes, bacteria or fungi in at the taxonomic level?
I feel like this is like a speed question round. No, this is really fun. So I'm actually so proud to say that one of the first things that I've accomplished since becoming the research theme leader in July 2020 was bringing together the soil spectroscopy lab and the soil living lab. So we specifically analyze for arbustral mycorrhizal fungi, AMF. And we do that because it doesn't need a lot of care in the soil samples. They don't need to be refrigerated or kept in, at a certain moisture control. And it can tell you so much about the health of the soil. We're also now growing bacteria and fungi. So we're looking at bacteria fungi ratio. And we have about 3 students from Kenya and 2 scientists in our lab just focused on this and trying to get as deep as they can in the taxonomic level, even species, but it's very tricky. And we're working with the National Museums of Kenya because this is a very understudied area that we're trying to fill the gaps. And even people who study soil biology, they usually don't do it at the landscape scale. So this is really exciting. And of course, we do earthworm assessments and, macroinvertebrates. Great question. Come help us.
Yeah, thank you. Probably I did not translate very well Dale's questions. He's more interested in how do you motivate individual producers or farmers to take part of your research? What is, and how do you convince them to be, collaborate with you?
Yeah, we always ask the farmers, what is the motivation to join? And you get all kinds of answers, but I think it's really interesting how much experimentation farmers do all the time, observation. And because it's a de-risking strategy, right? So instead of converting our whole farms in one practice, experimenting here. And I think our methods are very aligned with that. And the farmers that we work with are always asking for more co-learning events. So, sharing with their farmers, many farmers are now doing their own training events, training of trainers on their farm. We never hold workshops in hotels. We're always on farm, really digging in the soil and keeping ourselves really grounded. So, and farmers want to have more productive farms, and they don't want to just grow tomatoes like everyone else when you get a very low price and the market is flooded. So that diversification is really critical. So having water harvesting techniques that extend the growing season and having different tree crops are really important for a niche market. So it's really for, economic benefits, food production, and being able to provide for their family.
Thank you very much. Noel is asking again, are you familiar with the holistic management?
The holistic grazing management. So actually, we have a couple rangeland sites that we're doing the LDSF and looking at the influence of that bunching technique. So it's kind of following how the buffaloes are in nature. And so bunching the cattle to not overgraze areas and then move them, and also rotational bomas. So we're absolutely fascinated with that and doing quite a lot in rangelands. So Brent, even eCraft now is moving into rangelands.
Thank you very much. Julian, enjoy your presentation very much. Is asking, agroforestry is one of the many type of land managements. And how do you rationalize, incorporating other types of land forms or land management into your conservation, agriculture, and other aspects?
Yeah, exactly. It's such a good point, Julian, because that was my important point, is that we can't just look at one intervention. We have to look at the whole system. And so conservation ag works really well with trees. And there's a lot of examples of that here in Kenya. And climate smart agriculture, absolutely. Agroforestry has such an important role for sequestering carbon above ground and below ground and tapping into those deep soil resources. So, and also figuring out what are the best trees for that particular farming system is really important. And that's key to being able to see the scaling of such interventions.
Okay, I think we have one more question from Shelby. Again, enjoy your presentations, and she's asking how you handle the hesitant farmers, in your research, and whether there are any government, local, or municipal incentives encouraging farmers to participate in your research.
So it sounds like I need my sociologist right next to me to help answer all these tough questions. I'm going to remember that for next time. So for the question and answer, I have my friend, the hesitant farmers is a good way to describe it. So the early adopters are always the ones who will take up things first and fastest, but then in order to scale, we need to reach the non-early adopters, the hesitant farmers. And what we have found is that learning from farmers is the best way. So having these and facilitating these co-learning events, and showing the data, having the discussions with them. And we have a project that ended, this one I was highlighting, it was over 2 years ago. And the farmers are still going. They still have their groups. They're still going. The community facilitators are still there. It's being scaled. We keep getting evaluated. Everyone loves evaluating our projects because they're still going. And they keep asking, what is the motivation? They're saying, well, look, I'm growing maize in the dry season, or I'm having trees producing. I have so many crops, eggplants, and diversification. So I think it's really just co-learning from your peers, as we do in science, right? We co-learn from our peers.
Thank you very much, Leigh. I think this is sort of concludes our question answer period. I think we should stop it. And then I will again appreciate it for your very enriching and enlightening presentation on this topic that I wasn't really so much familiar with that. Now I'm going to turn over my colleague Guillermo to lead us through the research presentations.
Thank you, Nadir. I hope some of you can spare some time today for our student speakers. I invite you to stick around for the next 30 minutes as we lead you through some exciting and fast-paced presentations. You might find your next colleague here today. For those of you responsible for hiring the best and the brightest, you will hear from some excellent prospects today who will be graduating in the next year or so. Each of our students will have 3 minutes to present 3 slides. I will introduce each one, and if you have questions or comments, please feel free to place those comments in the Q&A box. Our students will be able to type any answers during the following presentations. Our first speaker is Maya Pejix.
All right. Hello, everyone. So the first part of answering the question of can agriculture be net zero in Canada was looking at a systems-based approach. A system-based approach is one which considers all the interactions between the components of a system and how this influences emissions. The systems-based approach for nitrous oxide emissions is pictured with soil type, moisture, crop type, etc., all influencing the amount of denitrification that occurs within the soil. This will then impact how effective different practices like no-till and nitrification inhibitors will be. Part of my research was finding out these interactions to better understand how emission reductions can be aggregated across Canada. After this research, I quantified and scaled up some of these greenhouse gas reducing practices to provide estimates of megatons of possible emission reductions. These range from less than 0.01 megatons to 32 megatons of carbon dioxide equivalent. These are by no means rigorous values, but they are an interesting aspect to consider. With greater systems being mapped out and understood, mitigation strategies can be more thoroughly scaled up. Lastly, intercropping provides an excellent opportunity for further systems-based analyses, as better understanding of interactions between different factors are needed to better provide general intercropping solutions. Next slide, please. The next component of addressing net zero agriculture is from a policy perspective. So an issue among Canadian agriculture is that certain commodities are just much more profitable than others. This factor needs to be considered when we're developing policies. The best implemented policies have the features listed on this diagram pictured. They are simple, they consider consumer demand, they provide autonomy for farmers, are cost effective, are governed by farm systems, take the regional environment into consideration, and lastly, include producers and their input into developing the policies. Payments for ecosystem services have been implemented in Ontario, Quebec, and Prince Edward Island. And some considerations for developing future payments are that complicated programs are less likely to be adopted. Different regions have unique environmental concerns, and careful planning is necessary to ensure that the policy doesn't cause farmers to intensify their existing production as a result of allocating land for these ecosystem services. And on to the last slide, please. And in order to reduce greenhouse gas emissions from agriculture, a greater system understanding, effective policies, and a change to the market systems are needed. Pilot net zero farms are a great way to put research into action, and one such egg farm exists in Alberta. If net zero emission agriculture is possible in Canada, it's only going to result from major changes in current practices. These shifts can happen over a long period of time, though, and current known practices can reduce emissions to some extent. Because of this, it is not likely that we'll see net zero emissions in the near future, but with considerable planning, it may be possible in the next 50 to 100 years. That is all for my presentation, and for more information, you can visit the website that I made, zeroag.ca.
Thank you, Maya. Our next speaker now will be Adam Fast.
Thanks, Guillermo, for the introduction. So Canadian Western Red Spring Wheat is the most widely grown wheat class in Western Canada. About 23 million acres were grown last year, 7 million or so in Alberta. And for spring wheat, nitrogen is typically the most limiting plant nutrient, and it's typically applied as granular urea fertilizer. During urea fertilizer applications, nitrogen can be lost to the environment and therefore reduce plant N uptake. Enhanced efficiency fertilizers have been developed to address this issue, and my research focuses on using EEFs with different nitrogen rates to optimize Canadian Western Red Spring Wheat yield and protein. Next slide, please. So here's a diagram depicting the typical fate of unprotected urea fertilizer when it's applied to field crops and how EEFs can function. Upon soil contact, urea first hydrolyzes into ammonium ions, and these ions, if they're near the soil surface, can be lost as volatilization. After this, the ammonium ions are converted to nitrate anions through nitrification, and due to their high solubility in water and negative charge, these ions can be lost to runoff or leaching, or used in the microbial respiration process of denitrification. The three most common types of EEFs are pictured in here. We have on the left, slow release fertilizers, which are designed to slowly release soluble fertilizer at a rate which matches crop demand. In the middle, urease inhibitors inhibit the enzyme urease, and nitrification inhibitors suppress nitrifying bacteria populations. The pictures on the left simply show conventional urea on the bottom, along with two EEFs to show you the visual differences that accompany each of these. And then on the right is an overview of my research experiment. We're growing one modern high-yielding cultivar, AAC Viewfield, using 6 nitrogen forms, and these are applied at 4 nitrogen rates. Next slide, please. So cumulatively, using my 3 years of data, I found that nitrogen form is not affecting yield of protein in wheat, but nitrogen rate is. The graph on the left shows that yields are significantly improved when using 180 kilograms of nitrogen per hectare relative to 60 kilograms, while 120 and 240 are producing similar results to both groups. Additionally, on the right, we have a protein graph showing how protein increases significantly with each increasing nitrogen rate. So far, these results are supporting the continued use of conventional fertilizer at rates of at least 120 kgs per HA. However, yield parameters are not negatively affected if fertilized with any enhanced efficiency fertilizer. Thank you.
Thanks, Adam. Our next speaker is Kia Light.
Hello, everyone. My name is Kia. Did you know that up to 70% of applied nitrogen fertilizer is lost to the environment, resulting in the pollution of our air and waterways? Environmentally smart nitrogen fertilizer, or ESN, could allow for increased nitrogen uptake and decreased nitrogen losses. Unlike conventional urea, ESN has a polymer coating which allows for nitrogen to be more gradually released. Water must first diffuse through the coating, dissolve the urea in the granule, which must then diffuse out of the polymer coating. This gradual release could allow for nitrogen to be available when the crop needs it most, resulting in increased nitrogen uptake and decreased nitrogen losses. However, there are many factors that influence nitrogen release, such as different environmental conditions and management practices. So how do we determine if ESN can increase crop nitrogen uptake? Next slide, please. We could do field experiments, but they may not capture the variability of different conditions, and they can be time-consuming and costly. Instead, computer models which can accurately represent ecosystem processes, such as EcoSys, can be used to determine if and under what conditions ESN can increase nitrogen uptake. First, however, the model must be able to accurately model the timing and amount of nitrogen released from ESN. To determine this, I modeled a laboratory experiment by inputting the soil moisture, temperature, and soil properties used in the experiment into the model in order to compare measured versus modeled nitrogen release. Next slide, please. Results are modeled nitrogen release, which are the lines, and measured nitrogen release, which are the points, for environmentally smart nitrogen in the red and urea in the blue, as a percentage of the total nitrogen applied as shown on the y-axis, plotted against the day of the experiment on the x-axis. Every 7 days, the temperature was increased 5 degrees, and the experiment was carried out at 2 moisture levels, 50% and 75% field capacity. Compared to urea, environmentally smart nitrogen has an initial delay in nitrogen release. For measured nitrogen release, we can see the delay between the black arrows, and for modeled, we can see that the delay lasts about a week longer for environmentally smart nitrogen as compared to urea, as highlighted by the black boxes. And this delay is due to that polymer coating. These results will be used to model conditions in the field to determine under what conditions environmentally smart nitrogen could increase nitrogen uptake. When crops are first planted, nitrogen uptake is low. Therefore, this delay could allow for more nitrogen to be available later in the growing season when the crop needs it more. This could increase nitrogen uptake, leading to higher crop yields and decreased nitrogen losses, a potential win-win for producers and the environment. Thank you.
Thank you, Kia. Our next speaker is Kira Konayewagek.
Thank you, Guillermo. Hello, everyone. I'm Kira Konowalik, and today I'm sharing my project on how Pennsylvania oil and gas policy addresses abandoned and orphaned wells. So legally, an abandoned well is basically a well that has not been under any activity in a year or is unequipped, and an orphaned well is a well that has long been abandoned. The risks associated with these wells are poor plugging that results in leakage of greenhouse gases, environmental contamination that can affect the landowner's property or have more widespread effects depending on the level of leakage, and also the cost to fix this. In 2021, the Department of Environmental Protection, or DEP, in Pennsylvania had documented 12,096 orphaned and abandoned wells, as shown in the image here, where blue are conventional and red are unconventional abandoned and orphaned wells. However, they estimate Pennsylvania has around 200,000 undocumented wells. Next slide, please. So under Pennsylvania legislation, several issues are clear in regards to orphaned and abandoned wells. Mineral and surface rights are separated, therefore a landowner cannot readily prevent a well being built on their property. A well operator must purchase a bond before drilling, which would cover future plugging and restoration costs. However, there are caps to the amount for larger operators as shown in this table, so no operator would ever pay more than $600,000 for any number of wells. Additionally, the actual cost of plugging, not to mention restoring a well, doesn't always match the bond amount, as this is on average $68,000. Therefore, the extra cost falls onto the DEP if the operator cannot pay. Another issue in the legislation is that funding for the DEP's plugging funds can't be derived from the general fund. Instead, it's supplemented by $150 to $250 permit fees, which are not enough to cover the cost of all orphaned and improperly plugged wells. So the DEP lacks the funds to cover the cost of all of these wells. Another concern in the legislation is the allowance of extension for inactive wells, as shown in the image here, with blue being conventional and red being unconventional inactive wells. So inactive wells permits last for 5 years and they can be extended, plus they don't need to be plugged. So these are at high risk of becoming orphaned or abandoned. Next slide, please. So the DEP has plugged 3,090 wells in 2020, and the number is higher now. Additionally, Pennsylvania is eligible for $104 million in federal funding for orphan well cleanup this year. So these are good steps towards the orphan well problem. However, in order to properly manage these wells in the future, It's necessary to change the legislation that prevents the allocation of proper funds and responsibility to maintain, plug, and restore these wells. Thanks for listening.
Thank you, Kira. Our next speaker is Helen McDonald.
Hi there, thank you so much. My name is Callum McDonald, and I'm pleased to present one of my side projects involved in improving plant oil analysis. Plant oils have a huge economic impact, from food purposes to bioproducts such as biodiesel and bioplastic. The global demand for plant oils has been rising due to population growth and anthropogenic climate change. Conducting laboratory research is vital to improving plant oil production, but science can be expensive due to chemical costs, labor, etc., as well as produce excessive chemical waste. Therefore, we need improved laboratory protocols for plant oil analysis that are cheaper, faster, safer, and greener. I have taken one common plant oil analysis method and modified several aspects for improvement. It involves extracting oil from the plants, adding some chemicals, boiling in acid for several hours or overnight, and lastly, analysis of the oil data. Next slide, please. Here, you can see the results of several modifications of the oil analysis protocol on 4 key species: flax, millet, alfalfa, and canola. The changes we made included grinding up the samples, switching to a cheaper and less dangerous acid, increasing the reaction temperature, and eliminating several unnecessary steps. These graphs showed that the changes we made allowed us to complete the oil extraction in just 75 minutes or less for all 4 species, which is a substantial improvement over the several hours or overnight reaction required by established methods. Similar improvements were achieved for 5 other horticultural species, not shown here. Next slide, please. So in summary, we were able to achieve the fastest reported reaction time, reduce the overall amount of chemicals consumed, switch to a less dangerous acid, optimize the reaction length for a total of 9 plant species, and reduce both chemical cost and labor input. This cheaper, faster, safer, and greener method produces equally reliable results on plant oils. This will allow limited research budgets to be stretched farther, shorten timelines, and reduce chemical waste, thereby benefiting plant oil researchers, farmers, and the environment. My email is callum@ualberta.ca if you have questions or would like to request my protocol. Thank you for listening.
Thank you, Callum. Our next speaker is Danielito Dolet.
Thank you. Imagine you are transported into the desert with no source of water. Once you reach your limit, your body will immediately perform processes to maintain itself to protect you from dehydration, and this would likely result to increased difficulty in breathing and fatigue to conserve water. But lucky for you, you are mobile and may chance upon a water source. Unfortunately for plants, they are sedentary and rely on external forces for moisture application. And without intervention, drought can be highly detrimental to the plants, leading to yield losses. They must maintain both above ground and below ground parts or processes through various drought responses in which one part or process may be prioritized over the other, directly and indirectly affecting their growth and functioning. With this, I hypothesize that drought has a negative effect on these plant parameters and a consequent negative impact on soil health by affecting essential soil microbes, nitrogen availability, and soil enzyme. Next slide, please. Following a greenhouse pot trial, I have found out that this was not the case for all parameters. First off, leaf chlorophyll content increased under moderate drought, suggesting that nitrogen accumulated in the leaf to produce more chlorophyll units. This accumulation sends a negative feedback to the nodule to stop nitrogen fixation to conserve energy being used up in the process. And that brings about a question: has nitrogen fixation decreased? To measure symbiotic nitrogen fixation, I will be grinding tissue samples and perform N15 isotope dilution technique in the near future. Second, soil extracellular enzyme activity was found to have mixed responses. Where carbon and phosphorus cycling enzyme was not affected by drought, in contrast, nitrogen cycling enzyme decreased in activity. The tricky part with extracellular enzymes, they are secreted by cells for a specific purpose. Their activity is linked on how much they are secreted, which is highly dependent on the condition of the soil, specifically for available nutrient. And lastly, interestingly, soil available nitrogen appears to have increased following drought. This is linked to diminished nitrogen uptake and improved rhizome deposition, increasing the release of nitrogen compounds into the soil because cell wall permeability increases during drought. Rhizome deposits also contain nutrients essential for soil microbes. Thus, was there a knock-up effect on microbiome community? A microbiome analysis will be done to try and answer this question. Next slide, please. I want to thank my funders and these wonderful people for their support on this project. Thank you.
Thank you, Danielito. Our next speaker is Cameron Mcdowell.
Hello, everyone. My name is Cameron, and I'm building further on my mass timber capstone project, which focused on a feasibility study in the city of North Vancouver. And in that project, we looked into changes in the city of North Vancouver needed to make in order to make mass timber construction more feasible. And I wanted to replicate that study in the Edmonton context to see kind of how Edmonton is to adapt to mass timber. So kind of starting off, what's mass timber? So mass timber is an engineered wood, and it's made from gluing pieces of wood together. There's two main types. There's cross-laminated timber, and there's glue laminate. Cross-laminated timber is placed and glued perpendicularly and is commonly used to make floor and wall plates. It can span large distances and is made as long as need be. And then glue laminated, also known as glue lam, is a fabrication where wood is laid in the same direction and makes beams. And then seen in the bottom picture, the pieces are assembled like giant building blocks to create these buildings, and its modularity makes it possible to manufacture off-site and quickly assemble once it's brought onto site. And then an interesting fact, the lumber doesn't just have to be healthy, mature, or old growth wood trees. Wood killed off from the pine beetle can also be used. While that wood is damaged and becomes gray, kind of like the bottom picture, it doesn't have an impact on the structure. So it kind of gives a cool aesthetic feature as well and doesn't use healthy trees. And next slide, please. So in North America, we have lots of low density buildings and high density buildings, but few in between, which is known as the missing middle. And then the missing middle concept is the idea of middle density housing, such as duplexes, townhouses, mid-rises, being forgotten. So conventional building methods such as concrete and steel get more return on investment by building higher than 20 stories. So mass timber can be most successful in the low to mid-rise density, where previous conventional methods such as concrete and steel wouldn't have otherwise been successful. And the city of Edmonton zoning already allows for mass timber kind of construction. And with the two zones, RA9 and high density residential, or HDR, on the map in the highlighted area shows the current downtown area with the RA9 and the HDR zoning. And then both zones allow for buildings up to 60 meters, which is about like 12 to 15 stories or a little more. So it's right within the high range that mass timber can go up to. And then next slide, please. So the National Building Code of Canada covers all the rules on how construction should and shouldn't be done. It was updated in 2020 to allow for wooden buildings to be built taller than 6 stories and upwards of 12 stories in height. This allows for wooden buildings to be built higher than previously allowed. And transporting goods along distances can increase greenhouse gas emissions, and less distance traveled is better for the environment. And then as seen on the map, each purple dot or star represents a lumber company that is a member of the Alberta Forestry Project Association. So it doesn't have to travel great distances to get our lumber thanks to that. And Edmonton is a strong industry and potential as seen from the map. And the proximity gives Edmonton potential to use the nearby local resources. And it'll hopefully help increase jobs and industry with the new potential sector. And this presentation by no means a comprehensive analysis of mass timber. There are many still questions that remain, but I hope that this presentation can be a start to the conversation of building mass timber. So thank you.
Thank you, Cameron. Our next speaker is Leigh Ann.
Thank you and good afternoon, everyone. My topic today is about deforestation in Amazon rainforest, how to promote sustainability by engaging with environmental and economic pillars using cost-benefit analysis. Before I get into the topic, let me use a few sentences to introduce the importance of Amazon rainforest. The Amazon is a huge region that occupies 40% of the South American continent. It is the largest remaining tropical rainforest on Earth. It hosts about a quarter of the world biodiversity and offered a lot of ecological services to benefit local and remote ecosystems, such as providing photosynthesis, storing carbon, and regulating hydrological cycle. However, major disturbances such as agriculture, ranching, and logging as a form of deforestation, jeopardizing Amazon rainforests to achieve sustainability, as they could cause severe environmental degradation at a long-term scale. Next slide, please. In terms of economic values, understanding environmental effects of deforestation is not enough to create sufficient outcomes, as there are many local people and businesses located in Amazon. We need to know how environmental value contribute to economic. Research showed that an intact rainforest provides a maximum of $1,431 per hectare per year of indirect environmental benefits by providing genetic resources, climate buffer, erosion control, and nutrient cycling. However, logging as a type of deforestation contributes about $2.75 billion of the total revenue. But if we think about economic loss of deforestation, just talking about climate change, it will cause long-term economic impacts. But if we conserve Amazon rainforest, the economic value will be even more beneficial annually. Next slide, please. So we must do something to guarantee these ecological services can be conserved for future generation uses. Agroforestry, as one of the land use management, grow trees and shrubs around crops. Also, empowering local people, especially youth and women, are also important because youth represent the next generation and women are more involved with agricultural and forestry products. Improving technology to do less harm harvesting and replanting forests to conserve ecological integrity. They are all potential programs that could be used to achieve sustainable development. And luckily, there are many NGOs making these programs happen and have shown some success to do sustainable development in Amazon. Thank you for listening.
Thank you, Leah. Our next speaker is Prabhajillo Arora.
Hi everyone, my name is Prabhajillo Arora. I would like to first acknowledge that I'm currently located in Amiskwaci, Waskahegan, Edmonton, which is situated on Treaty 6 territory. This area is a traditional gathering place and home for the Papas-Jays Cree and Métis people, as well as many other Indigenous peoples. The title of my project is The Toxic Legacy of British Colonialism in Canada, Environmental Injustice Against Marginalized Communities. So to dive right in, my project engages with social and environmental sustainability as, and this is also my thesis, it looks at how environmental harms propagated through white privilege affect the well-being of marginalized communities in Canada. Now, what is white privilege? White privilege is described in this context as the unearned advantages and conferred dominance accruing solely from one's race. And one of the ways in which racism can occur is from invisible societal systems conferring dominance upon one social group at the expense of another. And as you can see from my title, the toxic legacy that I talk about refers to the fact that owing to white privilege, many Canadians have accrued unearned economic, environmental, housing, educational, and health benefits by virtue of colonial and contemporary government policies and practices. And they have been able to do so at the expense of the health and well-being of Indigenous and African communities in Canada. Now, the focus of my research is on federal and provincial governments, namely Ontario, Nova Scotia, and British Columbia, to reveal how Canada is complicit in neo-colonial policies and practices that enable environmental injustices against Indigenous and African peoples to materialize even today, decades after Canada's independence. Next slide, please. Now, the details for each case study that I looked at from the three aforementioned provinces are on this slide. And you'll notice that the most repeated word here is ignore. And why is that? Because the general theme for each case study is government neglect to provision of basic rights like clean water and good health, lack of meaningful consultation with Indigenous groups affected by development projects, and lastly, government violation of treaty agreements, laws, and Aboriginal rights, which are especially in place to protect groups from historic marginalization. Next slide, please. And why should you and I be concerned about this. That's because environmental racism and environmental injustice have far-reaching negative impacts on the health, safety, income security, housing and food security, and educational security of members of marginalized communities. The federal and provincial governments that we elect, their lack of action to ensure welfare of marginalized groups and failure to enforce Aboriginal and treaty rights demonstrate that Canada still practices neocolonialism. And improvements and alterations to the dearth of inherently racist public policies regarding environmental justice for Indigenous groups are urgently needed. So I hope you take something away from this and do your part. Thank you.
Thank you, Prabhajillo. Our next speaker is Wame Mpong. Do we have Wame? Are you able to unmute yourself? In this case, perhaps.
Yeah. Hi, everyone. Yeah. Can you hear me? Yeah.
Yes,
okay.
we can hear you, Wame.
Okay.
Go ahead.
Now, nitrogen fertilizer uses or the increase. So there's a need to reduce the nitrogen fertilizer that we use on our field, especially that now that we have increase in urea fertilizer prices. So effort must be made to increase nitrogen use efficiency. Now, how do we do that? We do that through agronomic strategies, biological or synthetic nitrogen inhibitors. We also have molecular techniques and the use of humic acid. So humic acids are produced from organic and synthetic sources, for example, lignite, peat, and humalite. So my research is focused on the use of humalite. So humalite is really naturally oxidized coal-like material that is found in Alberta, and they also contain more than 60% humic acid and also high soluble carbon. So my methodology was that the research was carried out in a typical crop rotation system in Alberta. So last year we planted a wheat, and this year we are going to plant canola. So the experiment was carried out at 4 sites in Alberta. So urea rate was used, that is no urea, half and full recommended rate, and also 5 rate of humalite was used. That is 0, 100, 200, 400, and 800, 800 pounds per acre. The next one. So the result is that we didn't find significant differences between St. Albert site and Kara site, but we had significant difference at BRG and also the Go site. But with that, the treatment results did not differ from that of the untreated control. Yeah, so the next slide. Yeah, we also found the active carbon and also respiration. And we also saw that the treatment did not have any effect on soil active carbon and also respiration. We also found out that the treatment only had effect on St. Albert site, especially the St. Albert site increased the nitrogen or the nitrate content in the soil. And in effect, we saw that the application of humalite did not have much effect on soil agronomic and also the quality parameters. So this year we're going to have canola in this cropping season. That will help us to really analyze or assess the effect of this humalite in Alberta. Thank you.
Thank you, Wame. Our next speaker is Danica Andreas. Danica, would you unmute? Yeah,
there we are.
That's fine.
Good afternoon, everyone. My name is Danica, and today I'm going to highlight some key facets on the sustainability of plant disease management versus disease control. In Canada, infections of various crops directly reduce crop yields and profits by about 11.3% or $3 billion annually. This in turn reduces the amount of available food for people, profits for farmers, and contributes to an unbalanced agro-ecosystem. Next slide, please. So let's take a look at some of the advantages and disadvantages of plant disease management and disease control. Plant disease management is an integrative, proactive approach that mainly focuses on reducing the rate of transmission or preventing infections entirely. It does so by following 4 main principles of exclusion to prevent disease introduction, eradication of a disease once it has been introduced but before full establishment, the protection of crops via biological and chemical control if necessary, and growing resistant cultivars to minimize disease incidence and severity. It utilizes methods that focus on long-term goals of soil integrity and environmental health. This method does have its disadvantages as it requires more initial repetitive economic inputs and increases in labor as fields are needing to be regularly scouted for any visible signs of disease. Alternatively, plant disease control is a much more reactive approach that utilizes the application of a single severe measure, but only in the event that an outbreak actually occurs. The goal of this method is to capitalize on current market trends and maximize potential profit on a yearly basis. Action is only taken when necessary, which may assist in saving money in the immediate future if there is no disease outbreak. A disadvantage to this method is that frequent application of these pesticides have a detrimental effect on the soil and surrounding environment, and persistent diseases may not be completely eradicated and resistant strains may evolve. Next slide, please. So, which one is better? Ultimately, the decision of which tactic to implement is dependent on the goals of the producers. All facets of sustainability should be considered when making decisions involving plant diseases, and consideration for the long-term future of the land should be evaluated. Thank you very much.
Thank you, Danica. Our next speaker is Abhijit Pathi.
Hello. Hi, everyone. Today, I'm going to present about how to predict algal biochar yield using a machine learning algorithm. So quick background, biochar is a carbon-rich material, which have application predominantly in environmental field. And the market size of biochar is expected to grow at a compounded annual growth rate of 10%, more than 10%. So for our study, we have chosen why algal biochar, because algae have a high lipid content and it is very easy to cultivate in all type of environmental condition. And it's a third generation biomass, and biochar produced from algae is algal biochar. Pyrolysis is a complex process involving several reactions, and to understand those processes, we used machine learning algorithm, so that this modeling intended to scale up the process and predict the algal biochar yield. Next slide, please. Yeah, in the methods, what we have done is we have collected data from previously published literatures. We then use those data to train our machine learning model. We fed the data, both input and output parameters. Then we have tested the model using various, we cross-validated the model. Then we tested the model using various parameters such as R-square and root mean square error values. So this is how the model is working. We have used extreme gradient boosting machine learning algorithm. So here it will form decision trees, subsequent decision trees. It will try to reduce the biasness and variance in the data, and it will, in the final train model, it will try to predict accurately what is the output. Here our output is biochar yield. Next slide, please. Yeah, coming to the results. First, we try to understand what are the important parameters that is affecting our algal biochar yield. So from our model, we came to know that temperature is having the highest impact, followed by carbon, composition of algae, then H/C ratio of algae, then ash content, and subsequently we have ranked those parameters based on their impact. Then we also try to understand how exactly the parameter is affecting the yield. You can see in the graph, with increase in temperature, the SAP value is decreasing. It means the temperature is inversely proportional to the algal biochar yield. The similar type of exercise we have performed with all the other input parameters. Then in the summary plot, what we have done is we have tried to combine the importance plot and SAP dependence plot. So that we can see and understand how the parameters is impacting and which parameter is impacting the most in a single plot. To conclude, we have explored all the input parameter combinations. We have 13 input parameter combinations. So we have explored to do 13 combinations using machine learning model. Our model can predict 84% of accuracy for biochar, algal biochar yield using the 5 parameters. Then we have also used various plots to understand the effects. We came to know that temperature is having the maximum impact, followed by carbon and ash content. Thank you very much.
Thank you, Abhijit. And that concludes our final presentation. Thank you all for staying today and to be able to support the students with your participation. I hope that this gave some idea of the fascinating and diverse topics that our students are researching and developing. We also would like to express our gratitude to the personnel who supported us to be able to deliver this presentation today, this webinar. We have Tia, Lalani, and also Christine, and other many people who have been able to contribute and support this effort. We appreciate that. And also we appreciate that all of you took the time to be able to join today and to hear from our special guest speaker who is still around with us, Leah, and who have been participating in this 2021 Bentley Lecture. This concludes our webinar, and have a great afternoon. Thank you.